We're back, and we're now looking live at the Plasmodium genome database at plasmodb.org. And before we turn to the question that we raised on trying to identify candidate vaccine targets for malaria, let me just provide a little bit of context that may be a little easier to see than what we'd seen before in those canned screen dumps. The first point I'd like to make is that the success of the Plasmodium genome database has been such that it has led to its expansion to encompass a variety of other organisms. The PlasmoDB project morphed into the apicomplexan genome database, APDB, which was itself expanded still further into the eukaryotic pathogen genome database, encompassing a wide range of organisms, not only apicomplexan parasites, such as Cryptosporidium and Plasmodium and Toxoplasma and Theileria, but also other species as well, such as Giardia and Trichomonas, which we won't be talking about today. This project is, in fact, just part of a larger bioinformatics resource center project funded by the US NIAID that includes several different genome databases from dealing with a variety of pathogens. And those of you who are interested in other pathogens might want to explore this further. Now, the purpose of having an overarching website for exploration not only of malaria parasites, but many eukaryotic pathogens is that there are a variety of questions that you might want to ask that extend beyond an individual species. And there are several ways that you can explore that. I should also point out that this homepage also provides, in addition to links to some of these other pages, other bits of information. You might, for example, be interested in tutorials that highlight some of the features we'll be talking about. And further down the page here, you can see links to individual tutorials, links to publications, workshops, exercises, and so on. We can run questions across a variety of these organisms. If we were interested in apicomplexan parasites, for example, we might want to take a look at metabolic pathway maps for these organisms. And in this case, we've taken annotations from the Plasmodium genome database, the Toxoplasma genome database, the Cryptosporidium genome database, and mapped those on top of the KEGG metabolic pathway projects emerging from database projects in Japan. If, for example, we take a look at carbohydrate metabolism and dive in further to see the glycolytic pathway, the key to this analysis is indicated up at the top, in which Toxoplasma is indicated in red, Plasmodium is indicated in green, Cryptosporidium in yellow, and human in blue. And so we can see by looking at the painting of this metabolic pathway that from top to bottom, all of these organisms are capable of carrying out glycolysis. Now, that might not sound very surprising, but we can dive in a little bit deeper and take a look, for example, at the TCA cycle. And now we see a somewhat different pattern in which the yellow bug, in this case Cryptosporidium, doesn't do this pathway. And indeed, that's the case. Cryptosporidium is an anaerobe which doesn't carry out, doesn't carry out oxidative phosphorylation. There are many other pathways we could look at if we were interested, for example, in some of those metabolic pathways which we now know are associated with the apicoplast. For example, pathways involved in the biosynthesis of steroids. We could see that purely from the pattern of gene presence and absence, the red and green organisms, Toxoplasma and Plasmodium, clearly use a different pathway for synthesizing isoprenoids than the blue organism, human. And indeed, this is the case. One of the most striking findings from the biochemistry of the apicoplast is that these parasites synthesize isoprenoid subunits via a xylulose pathway typically associated with chloroplasts, quite distinct from the HMG-CoA reductase pathway found in humans. Cryptosporidium does neither, presumably salvaging isoprenoid units, which it can use apparently to produce squalene, but none of these organisms is capable of converting that squalene all the way into cholesterol. So this is not a sterile biosynthesis pathway, but it's certainly a pathway for the production of isoprenoid precursors. And there are many other fascinating aspects of parasite biochemistry to take a look at. So let's return to the eukaryotic pathogen genome database and return further to the Plasmodium genome database that is the subject of our discussion today. Now, as you've already seen, we can explore this database in many different ways. We can look, for example, at individual genes, and we'll just take a look at a single gene listed here, the default gene on this pathway, a gene known as the apical membrane antigen 1, a famous gene in the world of malaria biology because this has been advanced as one of the leading vaccine candidates for malaria parasites. Although there are a variety of concerns about AMA1 which lead investigators to be interested in identifying other candidates that might also be worth exploration. We can see that AMA1 is present on chromosome 11, and as you've already seen in the illustration from a chromosome-based view, is a highly polymorphic antigen. Many dozens of polymorphisms known to be associated with this gene, and we can see what those polymorphisms are from different species. We can see, for example, that this particular polymorphism changes the coding potential, such that in the reference 3D7 strain, the nucleotide C corresponds to a proline, whereas in many of the other species on this list, many of the other isolates, C to T nucleotide polymorphism results in a proline to serine mutation. We can see user comments which have been entered providing additional information on these genes, links to a variety of other gene pages, protein features which have been identified, a variety of means, predicted structural information, proteomic data indicating that there's evidence for expression at the protein level, microarray analysis on several different platforms indicating that this gene is most abundantly expressed late in the intraerythrocytic life cycle, as one might expect for a gene that is present in merozoites, the extracellular stage that one might want to target in a vaccine that would be effective against the disease-causing stage of malaria parasites. Additional information from other expression studies, from knockout studies, sequence information that can be shown here, and so forth. But as we discussed earlier, the real power of this database comes not solely from viewing it as a catalog of available information, but as an opportunity for being able to ask your own questions. So, what kinds of questions can we ask? Here, under the Queries and Tools link, indicated at the upper left-hand corner of your screen, we can see a grid describing a wide range of questions that one might choose to ask. For example, we might imagine that chromosomal location was in some way informative for candidate vaccine targets. I'm not quite sure how that would work. It's not really clear to me how proximity to a centromere might be indicative of a good target for vaccine development. And so I'm not going to pursue that line of inquiry, but you might want to if you have reason for thinking that chromosomal location is informative for effective vaccine targets. Let's instead start with some more obvious kinds of approaches. We certainly would expect that a target for vaccine development would have to be antigenic in some way. And so here we can take advantage of extensively curated information that comes from the Immune Epitope Database project, whose research on other databases has been incorporated into this database. And we can look for genes that have been annotated with a very high confidence of antigenic production against Plasmodium falciparum. And what we see is several genes. 41 to be exact. Now that's a disappointingly small number. It includes the merozoite surface protein 1, which with AMA1 is also viewed as a promising candidate for antimalarial vaccine development, and a variety of other merozoite surface proteins as one might expect. But surely there must be more than 41 candidate targets in a genome of many thousands of genes. So let's modify this query to ask a different question. Ask not just those antigens that have a high confidence of immune reactivity, but those that have any confidence of immune reactivity. And now we come up with a much larger list of genes which may have lower, from which we may have lower confidence, but things that we might want to explore further. What else might we want to ask? We'll return to our query grid here and ask about other information. So we've asked about genes that show some evidence based on manual curation of being effective epitopes. We might also imagine that genes that would be effective targets for vaccine development would have to be expressed in the right place and at the right time. By in the right place, we mean presumably on the surface of an infected red blood cell or on the surface of the parasite itself, and we can gauge that information by looking at cellular location here. We know that signal peptides are likely to be involved in targeting proteins outside of the cell. So let's ask for all proteins in a malaria parasite that have a predicted signal sequence. And we're interested in this case in Plasmodium falciparum, although we could interrogate other malaria parasites as well. And when we ask a question like that, we get a list of many hundreds of genes, including genes that certainly wouldn't be at the top of anyone's list as a vaccine target, such as this pseudogene that's indicated here. Interestingly, as I look at this number, while we find many hundreds of genes, they're not that many hundreds of genes. I would have naively expected that for an organism that makes its living by secreting proteins to modify the host cell, using those specialized apical secretory organelles that we discussed in the first lecture of this series, surely more than 10% of the parasite genome would be secreted. What could possibly account for this shortfall? These organisms, remember, are eukaryotic organisms, and this brings us face to face with the bane of genome annotation in eukaryotic species, and that is the following: that while we are quite good at identifying coding sequence, it's quite difficult to identify every single exon that is encoded into that's translated into a protein in eukaryotic species. And that's particularly true at the extreme 5' end of the gene. The first exon is the most difficult to identify. And that in turn manifests itself as an inability to accurately predict signal sequences. So we can imagine expanding our search a little more broadly to identify more proteins. Let's imagine, for example, if we go back to the grid of questions that we've asked, that we might want to ask not only for proteins that have a recognizable signal peptide, but also for proteins that have recognizable transmembrane domains, anticipating that those that have a transmembrane domain without a signal sequence are probably proteins for which we weren't really able to recognize the signal sequence accurately. Once again, we will look only in Plasmodium falciparum. I'm not interested in proteins with 2 or 10 transmembrane domains. I'm really interested in proteins that have at least one. I don't care how many, at least one and no more than 1,000 transmembrane domains. And now we see a slightly larger number, actually about double the number, 1,700 proteins. Now, some of those proteins, so this will presumably include many of those proteins with signal peptides. Some of them will be secreted without a transmembrane domain. But it includes many other proteins that we have some confidence are associated at least with a membrane, although we have no confidence that it's associated with the surface membrane of those proteins. Now, those of you with sharp eyes may have noticed that over on the far left-hand end of the screen is a box indicated as My Query History. And this is a history of all of the questions that we've asked in the context of this session. We asked, first of all, for this individual gene, AMA1. Secondly, we asked for the for the high confidence epitopes, and here for epitopes with even low confidence, proteins with signal peptides or with transmembrane domains. And what we're really interested in from the standpoint of location is genes that have either a signal peptide or a transmembrane domain. And so I'm going to combine these queries using a combination here to look for the results of our search for a signal peptide, that is query 4, or a transmembrane domain. And the result that I get will be a much larger set, or somewhat larger set, of about 2,000 proteins that have either a signal peptide or a transmembrane domain. Once again, it includes many proteins that I don't think any of us would advocate as vaccine targets, the cytochrome oxidase genes associated presumably with the parasite mitochondrion. But we can see now the results of this query, a new question, which I'm going to rename just so I don't lose track of it. I'll just call this signal peptide or transmembrane domain, just so I don't forget about what the question is that I've asked. So we can imagine a wide range of other questions, and I would encourage any of you who have questions you would like to ask to explore this query grid for accessible questions that may be relevant to the ways that you choose to interrogate the database. We might also want to know about proteins that are not only in the right place on the surface, but also at the right time. Remember that intra-erythrocytic life cycle in which a parasite invades into an erythrocyte, sets up its home as a ring stage parasite, develops and metabolizes and grows as a trophozoite, finally emerging by assembling daughter parasites as a schizont before rupturing outside of the red blood cell to release merozoites. We might expect that if we were interested in a vaccine that targeted the red blood cell stage of malaria that's responsible for the clinical symptoms, we'd be interested in targeting those merozoites, a very short-lived form about which it's difficult to gather detailed information. We could ask, for example, for proteomic for protein data, looking for mass spec-based data evidence of expression on merozoites. And you may wish to explore that. The data sets associated with transcription, some of which were described in Joe DeRisi's iBio seminar on malaria, are probably more extensive and more comprehensive. So I'm going to instead interrogate the expression profile from transcript levels. And there are a number of queries that can be used against various different organisms using various different data sets. Since you may be familiar with the data set generated in the DeRisi lab, we'll take a look at that data here, looking at expression timing based on glass slide microarrays, although there are other ways that you can interrogate this data as well. Now, we've already seen from looking at the expression profile of AMA1 that the transcripts were most abundant towards the end of that 48-hour window of replication inside an erythrocyte. And that makes sense if you imagine that transcription is going to precede translation. And so we might imagine that in that stage of schizogony, proteins are is the most likely time to transcribe genes that are going to be translated for protein in merozoites. And so I'm going to ask for genes that are maximally expressed at, in the last third of that intra-erythrocytic life cycle. That is the last 16 hours. In other words, I'll be looking for genes where expression is maximal at 40 plus or minus 8 hours. I don't care when the gene is turned off, but I'm going to look for genes that are upregulated by fourfold. You can change these parameters if you wish, and that are reasonably abundant, let's say in the top 60th percentile of all genes in the genome. And now we'll run this query and presumably return hundreds of genes that are that fulfill those criteria. 600 genes in this particular question, 600 genes, and we can see if I stand aside the actual expression profile, this hypothetical protein shows indeed the pattern that we expect, maximally expressed towards the end of the erythrocytic life cycle in all three of these strains, symbolized by the red, blue, and yellow curves. All right. Are there other questions that we might want to address? Well, as a geneticist, I guess I would be particularly interested in taking advantage of some of the most exciting new data sets that have emerged from malaria parasites, from resequencing projects designed to assess the diversity of parasites throughout the world. And as a result of such studies, we can identify polymorphisms, single nucleotide polymorphisms, or SNPs, that distinguish one gene from another. And so we'll consider comparing any two strains of our choosing, and I'm going to compare the reference strain, 3D7, the strain whose complete genome was first sequenced, with a field isolate from Ghana, the Ghana 1 strain. And we can set our parameters in various different ways. We could ask, for example, for polymorphisms that are known to affect coding potential, or for the density of polymorphisms. Just to keep things simple, I'm going to ask for any gene that has at least 5 known polymorphisms. But once again, you may want to manipulate these parameters. And asking a question like this gives us back several hundred genes, including, as we might expect, the variant surface antigens, the PFEMP1 genes, that I don't think would be likely advocated as a single subunit vaccine, but certainly genes that are likely to be highly polymorphic. There are many other questions you can consider asking, and this has already become a fairly long session, so I'm going to just limit myself to one more question, a question related to the evolutionary biology of these parasites. One might imagine, if we were looking for candidate vaccine targets, that we'd be interested in genes that are specific to malaria parasites. So we can interrogate for genes across the range of life for where those genes are found. And we might imagine, as we scroll down to look at eukaryotic organisms and the apicomplexa in particular, that we'd be interested in genes that are found in Plasmodium falciparum, of course. And perhaps if we wanted to consider a candidate target with broad spectrum activity, we might want to look for genes that are also present in Plasmodium vivax, the second leading cause of malaria in humans. But we're certainly not interested in genes that are present in humans. And so I'm going to ask in this particular question for genes that are absent from humans, or maybe absent from mammals altogether. And running a question like this gives me a large fraction of the genome, a third of the parasite genome, which is distinctive in being present in Plasmodium falciparum. Most of these proteins we know nothing about, hypothetical proteins. So let's return to our now rather long list of questions that we suggest might be relevant to vaccine development. We've asked for proteins that have antigenicity. That was our question number 3. And I'm going to try to combine that information with the other questions I've asked. I'm going to ask for proteins now, not for the union of proteins with signal peptides and transmembrane domains, as we asked earlier, but for the intersection of these various different queries. I'm going to ask for genes that have some level of immunogenicity and also have either a signal peptide or a transmembrane domain. So that was my question number 6. We're also present at the right time, expressed abundantly in chysons, and were highly polymorphic, indicating diversifying selection, presumably under control of the immune system, and also showed this evolutionary profile that were present in these particular species. So this is a question that I've actually never asked in exactly the same way, although I've certainly run many similar sorts of questions in the past. And I can see that in this particular set of queries, I come up with a list of 23 proteins. Let me turn off this track in showing the expression profiling so we can see these a little bit better, and I'm going to display all of them on one page. And now we can ask a little more readily about the various proteins we've looked at. So let's scroll down the list. First on the list, just first alphabetically, is a hypothetical protein. It's a conserved hypothetical protein. Is this a vaccine antigen? I don't know. But my eye is immediately drawn to what you might think of for this computational experiment as a positive control. That AMA1 protein, the protein that is one of the leading vaccine targets for anti-malarial vaccine development. A number of other proteins, a guanylyl cyclase, a kinase, several other hypothetical proteins. It's hard for me to believe that a tRNA ligase would be a good vaccine candidate. But here's the second of my positive controls, MSP1, the other of these leading candidates for an intraerythrocytic or an erythrocytic stage vaccine, and several other proteins which have certainly been considered. This CLAG9 protein has been advanced, for example, as a candidate target for vaccine development. So my point here is not to argue that computational approaches considered in and of themselves are ever going to be sufficient for identifying successful vaccine targets. That would be as absurd as saying that we can identify the function of the apicoplast solely by using cell biological approaches of organelle purification without any biochemical or genetic characterization. But certainly, in a few minutes, sitting here at the computer, we've been able to filter the many thousands of genes in the parasite genome down to a rather short list, a list of 23 that includes both of our positive control antigens, AMA1 and MSP1. And I would imagine that if I were interested in vaccine development for malaria, I would certainly want to explore further the other 21 proteins on this list as a manageable set that might be worth exploring for candidate genes that may be as good as or even better than AMA1 or MSP1 as vaccine targets for anti-malarial development.